Hello and welcome everyone. In this podcast we're going to talk the larger concept of gametogenesis. A very general definition of gametogenesis, as I'll draw here for you, is a biological pathway that takes a diploid cell, so a 2N cell, 2 copies of every chromosome, and through the process of meiosis 1, meiosis 2, and then, let's write maturation here, we produce a 1N, a haploid gamete. Often we make 4 of these, so I'm going to say times 4. Some organisms will make more than 4, but we'll go with 4 haploid gametes for now. Now, these gametes here, remember, they are different than the original parent cell. They're different because of, well, a few reasons. We can go ahead and list some of these reasons. One is you go from a diploid cell and you take that down to a haploid cell. So you have half the chromosomal content. In addition, the process of meiosis generates a great deal of genetic diversity. The first area where we make this genetic diversity is prophase 1, due to crossing over. The second area is anaphase 1. Together, these two generate a great deal of diversity so that the number of possible gametes is somewhere around 8 million for humans. Over 8 million, well over 8 million. Now I'm going to talk about the gametes a little bit more. Gametes is a very general term, but in general they are the cells needed for sexual reproduction. In organisms like fungi, the gametes would be called spores. Fungi, like yeast, the one you work with in the lab in genetics, will take a diploid cell and make 4 spores. And then those 4 spores are used in sexual reproduction for the fungi. Gametes could also be oocytes. These are the eggs produced by females through the process of gametogenesis. And then sperm as well, produced by males for use in sexual reproduction. We will often say that to make spores, that's a process called Sporogenesis, and to make oocytes, that's a process called Oogenesis. In fact, why don't we write those down? Oogenesis. Genesis meaning to create, to make, and oo for egg. And then spermatogenesis is the process of making sperm. And I'll write it up here. I said it before, but it's sporogenesis. So let's think about the gametes that can be created. We can define them into one of 2 categories. They could be isogamous, or they could be heterogamous. Isogamous means that the gametes are morphologically similar. Whereas heterogamous means that the gametes are morphologically dissimilar. And now I want to show you a couple of pictures to illustrate what I mean by these 2 terms. So I'm going to switch to a PowerPoint slide and then we'll come back to the whiteboard. Alright, now that I've defined isogamous and heterogamous, I want to show some examples. Fungi offer a great example of isogamous. We have Aspergillus spore here. Each of these spores are a haploid spore that has been produced through the process of meiosis and then have matured to have this very thick outer coating to protect them. But inside of them is just a single haploid genome, 1 copy of all their chromosomes. Same thing here with Saccharomyces cerevisiae. They hold all their spores inside of a single structure called a tetrad. But each of these 4 spores look very similar to each other, but inside of each of these 4 spores is a single haploid genome. So just 1 haploid genome in each of these. even though they look morphologically the same, again, remember, inside the cells, genetically, they're quite different. And fungi don't have males and females. They do have different mating types. So, for instance, they have in yeast here, they have an alpha mating type and an A mating type. So 2 of these spores will be alpha and 2 will be A. Doesn't matter which ones they are, just 2 of each. So that when it does germinate and these spores are released, they can mate with their sister spores here. Same thing here. They have mating types as well. Now, you're probably more familiar with heterogametes, such as humans' sperm and egg. You can see, unlike the fungal spores here, these are morphologically quite different. The sperm has the head of it at the top here and then a long flagellum. Inside of the head of the sperm is the haploid nucleus. I can put one in here. And along the flagella is a cytoskeletal structure to help it move, but it's also where all of the mitochondria is, right along this base part of the tail. None of the mitochondria turns out to be in the head of the sperm. And we'll come back to that later on when we talk about mitochondrial inheritance. The oocyte, the egg, is also haploid, but you can see it looks much different and it's larger. In fact, proportionally, it's even much larger than what we see here between these 2 cells. The sperm has just been enlarged to see it a little bit better. So that's what heterogametes is. They look different. I also want to just take a moment here to talk a little bit about the fact that not all sperm look the same. Not all eggs look the same. But there are some really noticeable differences in sperm. The first example is what we see in the soil nematode called C. elegans. We'll work with that in the lab later on. But these are sperm, and the thing you'll notice is that they lack this flagellum. They are non-motile. Turns out that C. elegans are hermaphroditic, so their sperm are produced right next to their egg, so they don't need to swim. This is a particular species of Drosophila, which has the longest flagellum known. So this is the length of an adult fly, and here's the head of the sperm in here somewhere, and you can see how the flagellum wraps around here, all the way around here, and to the end here. So it's an extremely large flagellum, just to highlight some of the differences between them. Another thing I want to talk about is the fact that gametes can survive a very long time, particularly the spores seen here in these fungal species, but also the oocyte in animals. And this is just a quick figure here to give you an idea of how long these oocytes can last, or spores. Human oocytes can last about 40 years, over 40 years. African elephant oocytes, they can last over 60 years. Baleen whale oocytes, they can last almost 100 years. Plant pollen grains can last well over 100 years. I should draw a line here. We don't really know how long, but they can certainly a lot longer than 100 years. And yeast spores seem to be able to last a little over 20-25 years. So in addition to making these gametes and specializing them, it turns out that they're made to last a very long time. Okay, let's move back to the whiteboard and finish this discussion. Now, though these gametes can look quite different and they have different morphologies, the end goal is that these 2 gametes from opposite mating types or produced by opposite genders come together through fertilization to produce a diploid progeny. Now what I'd like to do is spend a little bit more time talking about spermatogenesis and oogenesis specifically. And then at the end of that, I'm going to talk a little bit about some unique features of plants. Let's begin with spermatogenesis. So we could define this by just saying it's the meiotic pathway in males that produces genetically distinct haploid sperm. So let's talk about this pathway a little bit, and then we're going to talk about the opposing pathway of oogenesis. So we can start off with the individual, and all the cells, remember, in this individual are produced by the process of mitosis. So let's write the word spermatogonium. Now they are an interesting cell, and I'm going to just draw it here as a circle. They are diploid, so they are 2N. They, as well as the oogonium that we'll talk about in a moment, are the only two cells in the human body, or in mammals, that can undergo both mitosis and meiosis. They undergo mitosis to generate fresh spermatogonium. Always have to have a constant supply of the diploid spermatogonium so that you can continuously go through meiosis to produce more sperm. So spermatogonium undergo mitosis to generate identical cells to repopulate the ones that are being used in meiosis. We're going to spend our time here though on meiosis. Okay, so once this cell commits itself to entering meiosis, it becomes a primary spermatocyte. This primary spermatocyte is still a diploid. The primary spermatocyte will undergo meiosis 1. At the end of meiosis 1, it will be a secondary spermatocyte. At this stage, it becomes haploid. At the end of meiosis 1, the cells that are produced are haploids. So these secondary spermatocytes are haploid. It then will undergo meiosis 2 and produce what we call spermatids. At this point, meiosis is complete. These, of course, are also 1N. And we produce 4 of these from every primary spermatocyte. So I'm going to put times 4. And I should also point, in fact, well, let me do this. I'm going to draw these just to kind of show them in this equal way. An interesting thing about this process is that each of these spermatids are of equal size. They have equal cytoplasm content. They're indistinguishable by looking at them. Now, of course, the DNA in them has undergone a lot of variation through this process, but the gametes at this stage still look quite similar. The next thing that has to happen is that these spermatids have to go through a maturation process. So I'm just going to write mature there. And through this maturation process, they will form sperm. And as you saw on that slide I showed you a few moments ago, these sperm don't always have a flagella like I'm showing you here for humans. Sometimes the flagella, like you saw in the Drosophila species, is very large. And other times, like we saw with the C. elegans, there are no flagellum because the sperm do not need to be motile. Now, this is the process of spermatogenesis, going from a spermatogonium to a mature sperm. I want to point out a few points here on the side, and that is the first thing. The first thing is that spermatogenesis is continuous from puberty on. Second, in a lifetime, a typical male will produce about 525 billion sperm. In each ejaculate, there will be about 40 million to 1.2 billion. So a question we might address at some point is, why is it that so many sperm are produced per ejaculate, and why are so many produced in general when so few are actually ever used? There seems to be some kind of advantage for that, and so we'll talk about that as we move on. Alright, the next thing I want to talk about is oogenesis. And in talking about oogenesis, we're going to point out some differences between that process and the process we just talked about, spermatogenesis. Okay, so let's talk about oogenesis. And we'll give it a short definition here. It's the meiotic pathway in females that produces genetically distinct oocytes. So again, similar as we said with the spermatogenesis, is in a human, in this case in a female human, all their cells are reproducing by mitosis with the exception of the oogonium. The oogonium is a diploid cell. Just like we talked about with the spermatogonium, they also will go through mitosis to generate new identical diploid oogonium. We'll put new oogonium. They also are responsible for making oocytes, so they also have to go through meiosis. Similar as we saw before, when they commit to meiosis, the oogonium remains a diploid for now, and it's called a primary oocyte. This primary oocyte will now undergo meiosis 1 to produce a secondary oocyte. It will also produce a smaller polar body. Now, for comparison's sakes, when we were talking about the primary spermatocyte, when it went through meiosis 1, it produced 2 haploid secondary spermatocytes of equal size. This is a little different. They are haploid, but only this secondary oocyte will be large enough to continue on. So the secondary oocyte will now undergo meiosis 2. Sometimes the polar body will also undergo meiosis, but not always. Let's focus on this haploid secondary oocyte. As it completes meiosis 2, it will generate a smaller cell and a much larger cell. These are still both haploid, but this one is too small to be used in reproduction. And so this is another polar body. This one here is the ovum, the egg that will be used in fertilization. It is much larger than the polar bodies and much larger than the sperm in most species. Let's say a few things about this larger cell here. One, it's used in fertilization. I'm going to abbreviate fertilization, just FERT. Disproportionately high volume of cytoplasm. This means it has a lot, I put an up arrow for a lot, of mitochondria and other organelles, but particularly mitochondria and maternal RNA. This is very important for the future fertilization process. When the sperm fertilizes this egg, the mitochondria in this egg will be responsible for all the energy needed for those early divisions of that early embryo. In fact, none of the sperm mitochondria get into the egg at all, and we'll talk about that later. In addition, all of these maternal RNAs, they'll be translated into proteins necessary for early embryonic development. So it's very important that this egg is as big as it is disproportionately large. A few other things I want to talk about before we move on to plants is that if you remember when we talked about spermatogenesis, that process is continuous from puberty on. With oogenesis, there is a timing difference. This process of oogenesis begins as a fetus, but it stops at meiosis 1. So I'm going to write fetus here, and in your notes, you're going to have a little bit more room than I am. Indicate that oogenesis begins in the fetus. I'll write it over here. Oogenesis begins as fetus and stops at meiosis 1 in humans. Other animals will have different places where they stop. Once the woman hits puberty, each month one egg re-enters meiosis as it's released. So this second meiotic stage will reinitiate oogenesis. So I'm going to write over here, restarts oogenesis. So I'll say at puberty, oogenesis restarts. and it won't finish meiosis unless it's fertilized. So I'm going to just draw this arrow here and say finishes at fertilization. If it doesn't get fertilized, it doesn't complete meiosis. There's some interesting reasons why it might do this, and hopefully we have a chance to talk about that. If not, I'll certainly talk about it in a future class. Now I want to write down some numbers here. If you remember, I talked when we talked about spermatogenesis, how many billions of sperm are produced over a lifetime, and then how many are ultimately used. In regards to oocytes, at birth, a woman is born with all the eggs she'll ever have. And at birth she has about, or I should say greater than, 1 million. Interestingly, at puberty, there's only about 400,000. She's lost 600,000 of them. And then of those 400,000, through the course of a lifetime, from puberty until menopause, she will only use about 400 ova. I always think it's quite interesting to think about why are so many produced, over 1 million, with the intention of only using 400. This takes an incredible amount of energy to make all these 1 million oocytes, with the intention of only using 400 of them. Alright, just something for you to think about. Okay, I'd like to close this podcast with a brief description of plant sexual development. And this will be a fairly brief description, but it's important to look at some of the terms that are unique to plants. Okay, so let's begin this discussion of plant sexual development by drawing a flower. You will no doubt be super impressed with my flower drawing abilities. This flower represents the sporophyte stage. It is a diploid, so 2N. Within this flower structure, we have both male and female parts. And they're going to make very specific spore types. First, let's talk about the megasporangium. This contains, and I'm going to just write it up here, the part of the plant, the female part of the plant, called the pistil. And within this pistil is the ovary. This megasporangium, which is a diploid, will produce what we call a megaspore. This will go on to form the female gametophyte. We might also call this the egg sac. Now within the male part of the plant, we will find the microsporangium. I'm going to do my best to write this without erasing everything I wrote here. That's microsporangium. Microsporangium. This is what you will find here is, as I said before, the male part of the plant, the stamen. S-T-A-M-E-N. Again, through this process of meiosis, we will take this diploid microsporangium and produce the male gametophyte. We could also call this the pollen grain. It's important for me to point out that both of these are haploid, 1N, and together they represent the gametophyte stage, as I wrote here for each of these. So we have the sporophyte stage, which is the diploid stage, and the gametophyte stage, which is the haploid stage. These two now will produce the necessary gametes, the sperm and the egg. They will fertilize each other and form a 2N, a diploid zygote. Through the process of mitosis, this diploid zygote will form the sporophyte. So for a quick summary, the megasporangium and microsporangium are tissues found in the sporophyte stage. They will produce the male and female gametophytes in the gametophyte stage through the process of meiosis. They will produce the egg and sperm. Together they will fertilize each other and form a diploid zygote, which through the process of mitosis will form the next sporophyte stage. Alright, that's all I have for this podcast. We talked about gametogenesis, specifically about spermatogenesis, oogenesis, and then finally plant sexual development. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.